State of Play Research Information Series. And for those of you on Zoom, you might see a message about the meeting being recorded. Please just click OK or agree, and that notification will disappear. So just before we start, I'd acknowledge country. So in the spirit of reconciliation, MND Australia acknowledges the traditional custodians of country throughout Australia and their connections to land, sea and community. We pay our respect to the elders past and present and extend that respect to all Aboriginal and Torres Strait Islander peoples today. And I'd like to acknowledge the Wiradjuri people on whose land I am and pay my respects to elders past, present and emerging. So tonight we'll be hearing from two researchers who are working out how to better use the drugs we already have, either targeting them better or testing drugs used from different conditions. We'll first hear from Anthony White at the QIMR Berghofer Medical Research Institute in Brisbane, and he'll tell us how he's been screening for drugs that may have therapeutic potential in MND. And secondly, we'll hear from Cara Vine-Pero at the University of Wollongong. Her research focuses on how to enable drugs to penetrate the blood-brain barrier and access the nervous system. As previously, each presenter will talk for about 20 minutes, and we will have a joint Q&A session once both presenters have finished. You can ask your questions through the Q&A function in Zoom, or if you're on Facebook, you can use the comment section for questions. We'll monitor both channels and collate the questions and bring them to the Q&A at the end of the session. So that's enough from me. So please, let's get on with it and take it away, please, Tony.
Okay. All right. Thank you very much, Gethin. And it's great to be able to speak at this State of Play seminar. So I'm going to talk about how we've developed new cell models to look at drug screening for MND patients around sort of immune-targeted drugs. So just a little bit of background to start with. Inflammation is where you have abnormal changes in the immune response in the brain or the spinal cord. And this occurs in a lot of neurodegenerative diseases, including motor neuron disease, or particularly in amyotrophic lateral sclerosis, or ALS. And the main drivers of this inflammatory change in the brain or spinal cord are microglia, or microglia, depending on how you like to call it. These are the only specialized immune cell that exist in the brain or spinal cord. There are other cells that do have immune responses, but they are sort of secondary responses to their other functions. Microglia are the only specialized immune cell. The microglia have a lot of important functions, I guess, because they are the only immune cell of the brain and spinal cord. They're sentinels, so so they monitor what's going on in the brain and spinal cord, and they respond to this in a number of ways, including as the warriors. They clean up infections from bacteria and viruses in the brain. They also nurture the neurons. They can release factors to help maintain healthy neuronal function, and they maintain a normal healthy environment. So they remove dead cells and aggregated proteins and so on that we don't want in the brain. So really, if you want healthy neurons, you have to have healthy microglia. And this has become something that's well studied in recent years, that originally microglia was sort of thought as a secondary cell type that weren't very important, but they are critical for the functioning of neurons in the brain and the spinal cord. And if anything goes wrong in the microglia, then you have a lot of problems. So there's a lot of studies showing that microglia contribute to the disease progression in ALS. They can release toxic factors, particularly cytokines that can kill neurons. They also release various chemicals that can cause other cells, such as astrocytes, to release toxic factors and kill neurons. So they can start a chain reaction that leads to a lot of problems downstream. They also have reduced protective functions. So one of the main protective functions is phagocytosis, and that's a name that means that they engulf material and break it down and get rid of it, like basically the garbage trucks of the brain. And if that is impaired, then obviously you get this buildup of waste in the brain, and that seems to be something that's impaired in neurodegenerative diseases, including ALS. But the problem is that we don't really have a full understanding of the role of microglia in ALS, and what we do know has come from animal models. And they have been very important in identifying a number of the roles of microglia, but the more we study them, the more we realize that microglia in animals, and particularly in mice, are very different from microglia in humans. And this becomes even more so as people age. There's even bigger differences between the microglia of animals and humans. So we really need to understand what's happening to human microglia in ALS patients. So the ways to do this, there's a number of ways, but they all have a number of disadvantages. For example, we can use autopsy material, but this only really tells us about the end state disease, not what happens early on. Biopsy material, which is obviously very hard to get. I'm trying to take a piece of brain or spinal cord from people with ALS. You can also look at fetal microglia, but there's ethical issues, and again, that's not during the disease process itself, and that's something that's very difficult to get results from. And now we have stem cell protocols, which are called induced pluripotent stem cells. This is the main method of generating stem cells these days. But the problem is that to generate microglia, it's still a very slow and difficult process, and you get quite a variability in the outcome. So while it's very good for looking at disease processes in a small number of cells from a few people with disease, it's very difficult to use that to screen drugs, because you need large numbers of cells from large number of people with the disease, and that's just not viable with using this process. And there's another process that's become known in the last few years, which is blood cell derived microglia. So microglia don't normally come from the blood. But what we've been able to do, following some methods that were published a few years ago, is to be able to generate microglia using blood cells. And so this is really just a routine blood sample. We can take a sample as anyone does from someone with ALS. And what we do is we separate what we call monocytes. So these are particular cell type, also called PBMCs, or peripheral blood mononuclear cells, by spinning the cells through a gradient of material that separates all the different cell types. And then we take out the cells we want, and then we add particular chemicals to them called interleukin-34 and granulocyte macrophage colony stimulating factor. Very long name, but these two compounds, but when we add them to the cells, after 2 weeks, we get mature microglia, the same as what you would get in the brain. And so if you look at these pictures here, the picture that I'm illustrating at day 14 is what you get with mature, healthy microglia, sort of a cell body in the middle and these branches coming out, which are called processes, which is quite different from the monocytes from the blood that they started from. And the green stain and the magenta stain are markers to show that they are microglia. And we get upregulation of genes that are known to be markers for human microglia or mature human microglia. We also see phagocytosis, and this is a term, again, that means uptake and removal of material. In this case, when we add particles that turn red when you're inside the microglia, you can see that they've been taken up by the cells. And they release a whole range of factors, the cytokines, which are signaling factors to tell other cells what to do. They release all of these. So this slide basically tells us that what we have here are human microglia, or monocyte-derived microglia, MNDMI, as they're sometimes called in the literature. So microglia normally exists in a resting state that is highly ramified or branched. So ramified is just a term for branching, where you basically have a lot of branches coming out, just like a tree with main branches and secondary branches and so on. If they become activated and they're responding to something wrong in the brain, they become what we call an amoeboid. So they pull in those branches and become more rounder. So this is a resting cell here in a healthy control with lots of branches. In an activated cell, in this rapid disease case, the branches are being mostly pulled in and the cell is responding. And we can use this as a measure of the way that the cells are behaving in the disease process. By measuring the length of the branches, branch length, the number of endpoints, which is really just the number of branches they have, and the junctions, so how often they branch, as in a tree. Again, we have big branches and smaller branches and twigs and leaves. So you can have triple branching and quadruple branching and so on. And what we see is that in people with ALS compared to healthy controls, that there's a significant reduction in the branch length. And there's also a significant reduction in the number of endpoints, the number of branches. So the microglia that we're generating from people with ALS are very different and quite impaired compared to those with healthy controls. We can also see that there are differences in the number of junctions, so that they don't branch as well in the ALS cases. Now, TDP43, or TAR DNA binding protein 43, is a protein that's been studied for a long time in ALS, and it's known to have a very important role in the disease. It normally resides in the nucleus of cells, of all cells. In the disease process, it leaves the nucleus and aggregates outside of the nucleus. Still in the cell, but it's outside of the nucleus of the cell. And it seems to have an important role in driving the disease. But what we found in the microglia is we see these aggregates that occur in the cytosol, exactly what's been seen before in other cell types, and all sorts of different types of aggregates. So they can form different structures. And this graph here shows the shift from the nuclear localization to the cytosolic localization in the ALS patients. So they have a lot more cytosolic TDP43, the nuclear TDP43. And this is the first time this has been shown in microglia from people with ALS. So it's a really sort of important finding. And you may be aware of the work from Julie Atkin. She may have spoken before, and she published this work recently on showing that DNA damage occurs in ALS and is associated with TDP43 mutations. And there's impairments to the repair of that DNA damage. And we looked at this in the microglia, and we found that there was increased DNA damage in the microglia, so that the purple or magenta stain is just a marker for microglia, and the green stain is a marker for DNA damage. So all these little bright green dots are indicating damage, which you don't see in the healthy microglia. But in microglia from people with ALS, you do see these dots indicating increase in the DNA damage. So again, we're seeing another new change that hasn't been reported in ALS using these blood monocyte-derived microglia. And as I mentioned earlier, one of the most important functions of microglia, or a protective function, is called phagocytosis. So this is where it takes up anything that needs to be removed from the brain or spinal cord. And essentially, it wraps itself around whatever it is, takes it into the cell, and then digests it, kind of like how we digest food in our stomach, using acids and enzymes to break it down. And because it becomes acidic to break that material down, we can use a dye that becomes fluorescent red when it enters that acid environment. So you can see here with this red in the cells, in healthy control microglia, they have lots of red. But in microglia from people with ALS, there's a lot of cells that have very little or no red at all. So they're not taking up any of these particles. And you can see this again from the graph. The ALS microglia are a lot less effective at collecting the particles compared to healthy controls. We can also look at this in a slightly different way and look at the number of microglia, or MDMI, monocyte-derived microglia, that have taken up the particles And again, we see a difference in ALS compared to controls. And when we look at people with different disease courses, so we have people with a slow disease course, intermediate or rapid disease course, there's quite significant differences. So those with a rapid disease course have a much greater impairment of phagocytosis, of taking up the particles, than what we see in slow patients. So this may be a marker of changes in disease course for people with rapid disease. So over recent years, there's been a lot of interest in generating drugs that target microglia for the reasons that I've just covered. They're very important in disease processes, and also in ALS, there's a lot of money being put into trying to generate compounds that will normalize the function of microglia or stop their toxic inflammatory responses. But the problem is that there aren't really very good models for screening the drug efficacy for people with ALS or MND. This can only really be done in animal models or animal cells in culture. Human cell models have been very difficult to do this kind of work in. So we're now starting to look at drugs in our monocyte-derived microglia models. And we started off looking at a drug that targets a molecule called CD22. It's a protein that sits on the surface of microglia. And when it's activated, it basically shuts down phagocytosis. So as I mentioned, phagocytosis is very important for protecting the brain from accumulated proteins and dead cells and so on. And that's impaired in ALS. And so CD22 is a very important modulator of this activity. And it was shown in a paper in a top journal a little while ago that if you block the activity of this protein, CD22, you can restore phagocytosis in brains of aging mice. And as I mentioned, we see the inhibition of phagocytosis in ALS. So we block the activity of CD22 with an antibody. Compared to the ALS cells, we see an increased phagocytosis, increased function of these microglia. Even in normal cells, you can improve their activity. But we're able to bring this back to almost a healthy control level by blocking the CD22 receptor in ALS microglia. And at the moment, we're testing a range of compounds that have a better potential of entering the brain than antibodies, which are very difficult to get into the brain, to see if we can get the same kind of response using small drug molecules that can enter the brain. But something we also need to remember is that microglia don't exist in a two-dimensional plastic dish environment that we grow them in the laboratory. And this figure here shows what happens when you take microglia out of the brain. So this ex vivo means that it's just been taken, microglia have just been taken out of the brain, and all of these lines here represent the genes expressed in the microglia. And then after 6 hours, 24 hours, and a week in growing in culture on a flat dish, you can see how this pattern changes dramatically compared to when it first came out of the brain. That means that the microglia are changing very dramatically from what they were when they first were taken from the brain. And when they looked at the genes expressed from ex vivo, that means just out of the brain, to in vitro when they've been grown in a 2D flat dish in culture in the laboratory, a lot of the changes that they saw between the two are genes that are very important for neurodegenerative diseases. And so we've been working on trying to generate a model where we can look at drugs in a three-dimensional system. So we can grow the cells in a 2D model, in a 3D model where you can see the microglia have a much deeper environment so that they grow more like they do in the brain, because they can grow in three-dimensional directions and not two-dimensional directions. And we find that when we grow microglia from people, including people with ALS, they grow a lot longer. They survive a lot longer in the dish. They're a lot happier, as you would expect from being in a 3D environment. They last a lot longer there. And they express increased levels of markers that tell you that they're much happier and they're more mature compared to the 2D environment. And as I mentioned before, we can look at the branch length and the endpoints and so on. So we see increases in the endpoints in 3D compared to 2D, whether they're from people that are young or older. The number of junctions where they branch, that's also increased in 3D. Interestingly, the branch length, we saw increases in the young microglia from young people, but in older people, we didn't see these changes, suggesting that maybe as we get older, this branch length starts to become impaired. And maybe that sort of sets up a situation where in diseases such as ALS, which don't start in childhood, that they become more of a problem later on. And so we've been looking at also the effects of drugs in 2D and 3D. And we started off with a drug called the Satin Nib. And this is a drug that is in the clinic and it's used for treating leukemia. But it's been shown initially to restore brain function in mice with symptoms of Alzheimer's disease, and more recently to also improve outcomes in amyotrophic lateral sclerosis, ALS models. So it's a drug that targets what we call tyrosine kinase receptors, and it seems to lead to an improvement or a modulation of the inflammatory response, leading to an improvement in models of ALS. And when we put this drug, I haven't got data here from ALS patients because we're still working on that at the moment. But what we've done is initially we've done this on Alzheimer's patients, and we're doing this on ALS patients. What we found is that when we put this drug in 2D cultures of AD patients, we found that it inhibited some of the most toxic cytokines. So TNF alpha and interleukin 1 beta, these are cytokines that when they're released, they tend to kill neurons. And so we're able to reduce the expression of these in 2D, as many drugs do in the literature. When we did the same experiment with the microglia from the same people in 3D, we lost that effect. So we didn't see the protective effect of the drug anymore. So that meant that we suggest that this drug in treating microglia in the brain may not work. So we think it's very important to, when you screen drugs in 2D, to take them into 3D and see what they're doing in a much more complex environment that the microglia grow in the brain. And we can extend this complexity to brain organoids. And so don't worry too much about what the top levels are, but what we have at the bottom level here is stem cells that can grow through a series of phases into a mini brain or brain organoid. And these little dots here are human brain organoids compared to a $2 coin in a dish. And we can grow these and add in the microglia that we've been talking about from people with ALS and see how they behave in this brain function. So again, even a more complex environment with all the different brain cells growing in 3D, and then add the drug in and see whether that still has the same effect. And this may be able to really improve the translational outcome of the drugs when you get to the clinic, rather than simply doing experimental screening drugs in 2D, go into the clinic and treat people and find it doesn't work, as we've seen for so many drugs in most brain diseases. And thanks to the support that we've had from MND Australia, we've been able to establish a system where we can take microglia, grow them from people with ALS and controls, screen a range of drugs now and look at the various outcomes from that, and also confirm the how these drugs act in a 3D environment, and then take this potentially into the clinic with a much stronger feeling that the drugs may have a effect when you get into clinical trials. And just finally, I'd like to thank the people who did all the work that I demonstrated here, Dr. Hazel Queck in my group, and Carla Cuni Lopez, who's a PhD student who developed all this work, and Vincenzo from Italy, who supplied most of the ALS material that we will look at here today. And certainly for MND Australia, MND and me in Queensland, And without the Cole Bambrick MND research grant, the NTI MND research grant, this would not have been be able to present it today. We would not be where we are. And now I will hand over to Cara to continue with her talk.
Thanks, Tony. Sorry, just bear with me for one moment. Okay. All right. So thanks for a fantastic talk, Tony. Hard to follow up on that one. I just want to take this opportunity to thank Gethin and Laura for allowing me the opportunity to present some of the exciting work that we're doing here at Wollongong on improving drug delivery across the blood-brain barrier for motor neuron disease, and the support of MND Australia as well for some of the work that I'll be presenting this evening. So as sort of Tony alluded to in his talk, one of the biggest challenges facing the effective treatment of diseases of the central nervous system, such as MND, is the blood-brain barrier. And the blood-brain barrier, as the name suggests, is a physical barrier that exists between the brain's blood vessels and the cells and other components that make up the brain tissue. So what we're looking at here is a zoomed in schematic representation of a blood vessel in the brain. And you can see that the red blood cells are moving through that blood vessel there. And along the walls of that blood vessel are indicated here in purple are endothelial cells. And all blood vessels in the body are lined with endothelial cells. And between these cells, there are gaps that let molecules out of the blood vessels and into the surrounding tissue. And you can picture in this image here that these people are representing the endothelial cells in blood vessels throughout the body. And you can see that there's gaps in between these people that allow those molecules to diffuse through. But the endothelial cells that line the brain's blood vessels are wedged extremely close to one another, as indicated here by this image, where we can see these people linking arms. And they form what we call tight junctions. And these tight junctions are highly selective and allow only small molecules, fat-soluble molecules, and some gases to pass freely through the vessel walls and into the brain tissue. So the blood-brain barrier is ultimately in place to protect us and to prevent harmful substances such as toxins and infectious agents from diffusing from the bloodstream into the surrounding brain tissue. And I guess you can liken the blood-brain barrier to a COVID checkpoint, where we only allow people to cross the border or from the blood into the brain if they've been identified as essential or if they carry a COVID permit. The blood-brain barrier is essential for maintaining brain health, but it has a downside, and that is that, when we're trying to treat diseases such as MND, the vast majority of drugs that we are developing don't readily cross the blood-brain barrier. So it poses a real big challenge. So an example of a promising class of drug that doesn't cross the blood-brain barrier, but it's under clinical investigation and it's not yet been approved for human use, are a class of drugs known as Antisense Oligonucleotides, or ASOs for short. So we know that MND is associated with protein misfolding and aggregation, and the accumulation of toxic protein aggregates in the upper and lower motor neurons of the motor cortex and the spinal cord over time is associated with motor neuron death. And this leads to the loss of muscle control, muscle atrophy, and paralysis, which is characteristic of motor neuron disease. But ASOs have been developed. These are synthetic or artificially created drugs that are a sequence of nucleotides similar to what sort of makes up our DNA. And they've been designed to specifically bind to these disease-causing proteins and reduce the levels of harmful protein that's being produced, prevent their aggregation and their detrimental effects on motor neurons. So a particular type of antisense oligonucleotide that's been developed targets a mutant form of superoxide dismutase 1, or SOD1, which you may have heard about. And these mutations account for about 20% of all familial or inherited MND cases, and about 2% of MND cases overall. And these mutations cause the protein to misfold as it's being made, which leads to the toxic buildup of misfolded protein, as I mentioned previously. So we can see here that all of the information required to produce this protein is contained within our DNA. And then it's transcribed into mRNA, or messenger RNA, which delivers these genetic instructions to the rest of the cell, where the protein can then be made. So the antisense oligonucleotides that have been designed against SOD1 are designed to specifically bind to this SOD1 mRNA, as indicated here by this pink strand. And it targets it for degradation, and it prevents this mRNA from being read, and therefore we don't get the disease-associated proteins from being produced. And by reducing the levels of SOD1 protein, we can slow the progression of this form of MND. So to date, the preclinical and clinical data has been really promising. So we've seen the injection of antisense oligonucleotides directed at SOD1 in mice and rats expressing this mutant SOD1 has reduced the spinal cord levels of the SOD1 mRNA and protein, as we expect. And similarly, in MND mouse models, a single injection has been able to delay disease onset, improve motor function, and increase overall survival in these animals. Whereby we see an increase in the median survival of mice treated with antisense oligonucleotides from 168 days, where we inject an inactive antisense oligonucleotide, all the way to 205 days when we inject the active version, which is responsible for stopping the production of that mutant SOD1 protein. This ASO treatment also preserved neuromuscular innervation and function. So some promising results, and you may have seen that there are clinical trials of this ASO in phase 1, 2, and 3 currently. However, a limitation of this technology, as I mentioned previously, is that antisense oligonucleotides are unable to cross the blood-brain barrier, and this means that we see reduced amounts of available drug in the central nervous system where it's required to act. Furthermore, the antisense oligonucleotide has a short circulation half-life, so it's quite easily degraded in circulation if we inject it into the bloodstream. It's broken down by nucleases in the bloodstream, and it's cleared quite quickly via the renal system, so via the kidneys, and excreted in the urine. And because these antisense oligonucleotides can't cross the blood-brain barrier, we need invasive routes of administration, for example, intrathecal and intraventricular routes of injection, in order to get the drug to the central nervous system. And this can be quite problematic. You know, in some of the clinical trials so far, we've seen some adverse events associated with this route of administration. And we also see post-surgery complications as well, which is particularly problematic when the patient has to receive multiple injections of these ASOs throughout their treatment course. So we need better ways of delivering these antisense oligonucleotides. So my lab has been focusing on new and improved ways we can deliver these antisense oligonucleotides, whereby we can protect these nucleic acids from degradation while they're in circulation, allow for cellular uptake into the cell types that we're targeting, as well as facilitating delivery across the blood-brain barrier. So what you're looking at here is a drug delivery system that we have developed based on a lipid nanoparticle, which is similar to the technology used in the Pfizer and Moderna COVID-19 vaccines. So here we load the antisense oligonucleotide, we couple it with calcium phosphate into this core region here, and we coat it with lipids or fats, essentially, some of which are naturally found within the body. These particles can't be seen by the naked eye, and they're about 1,000 to 2,000 times smaller than the thickness of a human hair. But essentially, as I mentioned, they allow the fragile drug to remain in circulation longer. And because of the lipid coating, we can also facilitate a certain amount of delivery across that blood-brain barrier. So we've shown that our SOD1 ASO-loaded nanoparticles are non-toxic to cells. And here we're just looking at cell viability across a number of concentrations. And we see that viability remains around 100% for these cells, that they're taken up into motor neurons. And so these are primary motor neurons here that have been stained in green, and our nanoparticles have been stained in red. We can see they're taken up into these cells, into the cell body and along the neurites there. And in addition, they maintain the antisense oligonucleotide maintains its activity. So here we're looking at SOD1 protein levels. So here, this is a negative control, the cell only. So this is just the baseline level of protein that these cells are making. And when we deliver our SOD1 antisense oligonucleotide via our nanoparticle, you can see that we can reduce the amount of protein that's being produced by those cells, but without causing any toxicity. So the cells are remaining viable throughout that treatment course. In addition, we've looked at the biodistribution and pharmacokinetics of our SOD1 ASO-loaded nanoparticles in mice. And so this is a busy slide, but what I want you to focus on here is, sorry, this is automatically changing the slides on me. So what we're looking at here are, in this particular panel, are the kidneys that have been taken from different mice after they've been treated with SOD1 antisense oligonucleotide that's had like a fluorescent probe attached to it, so we can image and see the location of that antisense oligonucleotide. And you can see as early as 1 hour that the kidneys are lighting up. This is showing us that the antisense oligonucleotide is being really quickly excreted via the renal system. We see again at 3 hours, those kidneys are lighting up as well. But in this treatment group here, where we've loaded the antisense oligonucleotide into our nanoparticle, we get reduced signal associated with the kidneys. And we know that this is because the nanoparticle is continuing to circulate around in the bloodstream for longer. And we've been able to quantify this, and you can see after 6 hours, we have significant reduction in ASO signal in the kidneys when it's in the nanoparticle formulation compared to when we inject just the naked antisense oligonucleotide. And this actually results in trends towards increased uptake of that antisense oligonucleotide in the brain compared to the naked antisense oligonucleotide. So while these results were promising, we really wanted to improve the efficiency of delivery to the central nervous system. And so we turned to a relatively new and exciting approach to temporarily open the blood-brain barrier known as focused ultrasound, or FUS for short. So FUS is a non-invasive technique that uses acoustic waves in the ultrasound spectrum to selectively and focally disrupt the blood-brain barrier. So you can see here we have the ultrasound transducer on the patient's head, and we can pass those sound waves harmlessly through the bone and tissue, and they converge on the targeted region here that we call the focal point. And you can liken this to using a magnifying glass to focus the rays of the sun to a point. We can target virtually any region in the brain with millimeter accuracy using this particular technique. And when we inject tiny gas-filled microbubbles into the bloodstream, these microbubbles circulate through the body and end up in the blood vessels around the brain. And when these microbubbles come in contact with these sound waves, it causes these microbubbles to oscillate or move around rapidly. And this imparts mechanical forces on those endothelial cells that I was talking about and temporarily breaks those tight junctions. And this allows the drugs to enter from the bloodstream into the brain tissue. So studies have already demonstrated that the safety of focused ultrasound for blood-brain barrier opening in patients with brain tumours, as well as Alzheimer's disease. And recently, the results of a phase 1 trial looking at FUS to open the blood-brain barrier in MND were recently published in 4 patients and showed the safety of this approach. But no one has really looked at combining the use of focused ultrasound and microbubbles with drug delivery systems in the context of MND. And that's something that my lab is doing for the first time. So I'll talk you through those results now. So we initially conducted a study in mice to determine the optimal parameters that we needed to safely open the blood-brain barrier in mice. And we used MRI guidance to allow for the dynamic feedback of the extent of blood-brain barrier opening via the measurement of a contrast agent known as gadolinium-3 leakage into the brain. So ordinarily, this contrast agent doesn't enter the brain, but when we've compromised the blood-brain barrier, we do see it show up on T2 weighted MRI. So what we're looking at here is coronal, transverse and sagittal images of the mice before we've treated with focused ultrasound. And then here we're looking at those same viewpoints after focused ultrasound. And here we can see there's negative contrast in these brain regions, which is associated with gadolinium uptake and therefore blood-brain barrier disruption. What we found from these studies that the focused ultrasound parameters that we did use was well tolerated in the mice and that the opening of the blood-brain barrier was temporary. So you can see here in this particular study, we've applied focused ultrasound. We see that gadolinium contrast emerging in the brain here on day 0, but we're seeing a reduction in that contrast agent over time. And by day 5, we see complete recovery of that blood-brain barrier. So it is reversible. So next we wanted to see if we could enhance the delivery of our antisense oligonucleotide loaded nanoparticles into the brains of these mice when we applied focused ultrasound. And so what we're looking at here are mice that have been injected with the nanoparticles. And again, we have a fluorescent recorder or probe so we can track the whereabouts of these nanoparticles in the brain. And in these mice, they were injected with the nanoparticles. Focused ultrasound was applied, but the microbubbles weren't injected. So we didn't have anything available to mechanically disrupt those endothelial cells. And so we see no contrast agent here by T2 weighted MRI, and we see no signal in the brain associated with our antisense oligonucleotide loaded nanoparticle. Conversely, when we injected the nanoparticles, we applied focused ultrasound and we also injected those microbubbles, we saw evidence of blood-brain barrier disruption. We saw evidence of ASO nanoparticle uptake in the brain and specifically in the region where the blood-brain barrier disruption was noted via MRI, which was really exciting. We quantified this and found on average that we could increase the penetration of our ASO-loaded nanoparticles into the brain by 4 times compared that of our controlled mice, which was really exciting. And we further showed that the extent of blood-brain barrier opening directly correlates to the amount of nanoparticle uptake in the brain, as indicated by an increasing signal in the brain. So what we're looking at here is low, medium, and high focused ultrasound mediated blood-brain barrier disruption, and that directly corresponds with the ASO-loaded nanoparticle signal in the brain here, and we quantified that and saw a really nice linear correlation between gadolinium uptake in terms of nanomoles and fluorescence intensity from those nanoparticles. So where to from here? Well, in order to translate, these exciting findings to MND patients, we next need to demonstrate that our focused ultrasound mediated delivery of our ASO-loaded nanoparticles is efficacious in disease models of MND. So what that means is that we want to show that we can improve survival in those mice that we're treating with ASO nanoparticles and FUS compared to those mice that don't receive FUS. And we can maintain muscle performance and other clinical features associated with the treatment. If we can show this, it would be a significant breakthrough in the field, we think, because we're the first people to ever combine these two different strategies together. And while we're sort of focused on delivering antisense oligonucleotides directed towards SOD1, we could also load other antisense oligonucleotides directed at C9orf, for example, and even TDP43 that Tony mentioned previously, the latter of which accounts for about 95% of all MND cases. So there's real potential to have huge impact on the field if we can demonstrate that our approach is feasible. We could also use our technology and the combination of our focused ultrasound and nanoparticle approach to improve the bioavailability of existing therapeutic strategies, so ones that are currently approved for use, for example, riluzole. We could try and improve the uptake of this agent into the brain beyond its current uptake, which is quite minimal. We could also apply this to new drugs that are emerging from drug screens, like the ones that Tony's conducting, and ones that are even in clinical development as well. And then finally, we know that MND results in the destruction of upper and lower motor neurons in the motor cortex and the spinal cord. And while we've demonstrated that we can use focused ultrasound to open the blood-brain barrier and enhance drug delivery, we also want to look at what parameters we might need to open the blood-spinal cord barrier, so we can target the anterior horns of the spinal cord as well in combination. So this is something that we are looking to pursue in the near future. So with that, I'd just like to finish up. Thank my amazing postdoc Gayatri, pictured here, for generating all of the ultrasound, focused ultrasound and MRI images that you saw this evening, and my previous lab research assistants, Lee Yu and Claire, for doing a lot of the workup of the nanoparticle technology that I presented this evening. My collaborators, Justin Yerbury at University of Wollongong, Chris Thorect at the University of Queensland, and again, really grateful and thankful for the support from MND Australia for funding some of the work that I've presented here this evening. So I'm happy to answer any questions.
Great. Well, thank you both. Thank you, Cara and Tony. And we'll go into a few questions now. Tony, if you'd like to, yeah, great, you're back online. So a few questions here. With the focused ultrasound, I might have missed this or I might have got a miss, but has this been tested in humans? Are there drugs already out there that, or sorry, treatment approaches that are already using this, or is it still very much in phase 1?
So it has been tested in humans. So when we've seen the application of focused ultrasound in conjunction with systemically administered chemotherapy, for example, to target certain brain cancers. The feasibility and safety has been assessed in MND patients, in 4 patients in a phase 1 clinical trial. whereby they're not administering any therapeutics, but just applying the focused ultrasound to look at tolerability. And to date, that's been proven to be safe. And there's some researchers at the University of Queensland, Professor Jürgen Goetz, who's also looking at the application of focused ultrasound in Alzheimer's disease to clear plaques. And my understanding is he's conducting a phase 1 clinical trial at the moment using this particular technology.
Okay. And you mentioned using, other than SOD1, say C9orf and TDP43. Is it viable to put all those into the same nanoparticle or treat people for, say, all 3 genes at the same time? I've no idea if that's even a reasonably medical proposition, but is that possible if you think you could knock down a bunch of genes in the same pathway or the same at the same time?
Yeah, look, I think first of all, you'd need to, if you're looking at familial cases, you'd want to look at the genetic drivers of the disease and target it that way using your antisense oligonucleotide. But, you know, I think I've got a background in cancer therapy as well. And I mean, combination therapy really is the key. So if we can, as you say, target multiple pathways involved in driving disease, then we're more likely to see an increased or improved therapeutic response than targeting a single pathway. I know in the context of cancer, for example, if you're targeting one single pathway, you may respond initially, but then later the patient relapses. And this is because there are other pathways that come into play and you start to see a resistance to that therapeutic. So if we can hit multiple targets, we're likely to see a better response.
Thank you. Question for Tony. So with the 2D versus the 3D system, is the 3D system a viable sort of first screening approach rather than the 2D? Because you're suggesting that basically perhaps the 3D is more representative of what happens in the body, and therefore is it even worth screening in 2D if you have 3D options?
Yeah, that's a good question, and we don't really know the answer because we haven't screened enough drugs to say how many that work in 2D will work in 3D. So we've seen from some examples that there is a big difference there, but that may not be the case for a lot of drugs. They might work just as well in 2D as 3D. Any kind of 3D model, whether it's what we're working with or organoids and so on, despite what people say, and you often see this all the time, when, you know, we're growing brain organoids and we're going to screen drugs, you know, large numbers of drugs and so on. That's something that we're still a long way off being able to do, screening thousands or, you know, hundreds of thousands of compounds in organoids and 3D models. But we can whittle the number down using 2D systems and look at promising targets in 3D. And then if they get through that system, then it's really about just doing everything we can to improve the outcomes when you get into clinical trials. And that's using mice, using 3D, 2D, everything. If you can find drugs that work in all the systems that you look at, then it's you know, going to have a much better chance compared to the very poor success rates we've seen so far.
Okay. And what you listed a few drugs that seem to be targeting microglia. Have any of these been tested in MND yet?
In the patients,
yes.
No, not at this stage. So we're also working with some other groups that have drugs that they're interested in putting in patients. Obviously, a lot of things have stalled because of COVID, and that's been a bit of a problem. But we have a number of drugs that we're looking at that could end up in clinical trials once things get up and running again.
And what about the blood-brain barrier then, bringing the two sort of approaches together? How do you, are all the drugs that are targeting microglia, do they all cross the brain-blood barrier easily enough?
As I mentioned, looking at the anti-CD22, that's something that we don't know whether enough of it crosses the blood-brain barrier. So we're looking at small molecule drugs that that will cross the blood-brain barrier. It's well known what types of drugs can penetrate the blood-brain barrier. So if you've got something, one of those sort of classes of drugs that works in your model, then it will get into the brain. But we're actually doing the same kind of work that the CARA is doing in animals. We're also doing that with the FUS in our cell culture models as well, and we're collaborating with CARA on that. So we hope that we will have some sort of pipeline system where we can look at drugs in cell models and animals using things like FUS to increase the delivery, the ultrasound to increase delivery of the drugs.
There's other questions here about the blood-brain barrier. When you open the blood-brain barrier with microbubbles, is there a problem that other compounds can also penetrate that shouldn't perhaps be there?
Yeah, look, I think while that blood-brain barrier is open, then essentially anything that's in circulation can cross during that time. And so I guess the key is finding the balance between how long we can disrupt it, whether or not we can get our drugs to therapeutic concentrations in the brain, and then how long it takes to quickly close again. There's lots of studies that are ongoing now looking at, neurotoxicity or neuroinflammation with blood-brain barrier disruption and trying to kind of walk that fine line. So we're learning a lot more about it along the way. But I think the most important thing is that to date, we've seen, tolerability of this approach in humans. And so that really kind of gives us confidence that, what we're designing and developing here is going to be translatable to humans at some point, and it's safe and tolerable.
Another question here, are there any drugs or even experimental drugs that are really not suitable for this approach, trying to get them across the blood-brain barrier? Are there drugs you're just thinking they're not going to work?
I guess something that you need to consider is once you've disrupted the blood-brain barrier, the drugs still need to penetrate into the brain parenchyma, into the brain tissue. And we know that certain drugs do that better than others. For example, drugs that are fat soluble or lipid-like will be able to penetrate further than drugs that aren't, for example. And smaller drugs will be able to penetrate further than larger drugs. So, there's studies ongoing, with antibodies, for example, but there may be limitations with using such large macromolecules because they can't penetrate far enough away from those blood vessels to reach the regions where they're actually required. So I don't know whether Tony had any more to add to that.
No, I think as you covered, there's a lot of studies showing that you can open the blood-brain barrier safely, but there hasn't been a lot of examination of what drugs will get into the brain. What some groups have found, and we're doing some work in this area as well, is that there seems to be a lot of complicated things going on when you use the ultrasound, and it may actually also help some drugs to be transported across cells into the brain, and it's not just about opening the brain barrier, but it actually improves transport as well. We're also looking at drug transporters. Some of these are increased, some are decreased, so you might get more uptake of a drug and less efflux of a drug from the brain. So it's quite a complicated system, and we're trying to sort of tease those areas apart. But it does look very promising in terms of maybe not every drug will get in, but I think there's going to be a large number of drugs that will benefit from this process.
Right. And is there a trial at the moment with the focused ultrasound and riluzole that you're aware of? Is anyone testing that?
No. The last clinical trial with focused ultrasound in the context of MND was the phase 1 trial, and I think they published those study results in 2019. As far as I'm aware, there's no recruiting trials looking at the combination of therapeutic delivery and FUS. So I think it's an area, you know, that we will start to see more activity in as we learn more about the effects of FUS on those cell types in the brain. Yeah, I guess people are sort of turning to looking at combining that approach with therapeutic delivery.
Thank you. And Tony, so is there any data around how early in the MND progression you get changes in microglia? Is it thought to be, is it chicken and egg type thing? Is it, you think, driving disease, or is it in response to whatever triggers the disease?
Yeah, it's a good question. It's hard to know, because in animal models, it seems that they're in the early stages of disease that there's microglia are become upregulated, and they may have a sort of protective response. But as the disease goes on, they sort of lose that protective action and become more toxic. But as I mentioned, what we're finding is that there's a lot of differences between what microglia do in animals compared to humans, and we don't really know at this stage what the changes are across the disease process. We know that we can see a lot of changes to microglia, but trying to understand the exact process of that over the disease course is something we don't have an understanding of at this stage, and we're trying to work that out. And something else that we found is that often microglia can do the opposite in humans than what they do in animals, particularly as we age. Microglia in humans tend to become overloaded and they age themselves, and they become less functional with age. They don't carry out their functions correctly. Whereas in animals, I guess probably because they only live for a few years, that the microglia tend to be okay throughout that disease process. So microglia, we think that they survive in the brain for a very long time in humans, and they do become sort of worn out from the job they have to do. So these are kind of differences that we're trying to tackle at the moment, and understand what happens as we age, and how that affects microglia, and then you add the disease processes on top of that.
Great. And just one last question for Cara. So obviously it'd be great to get away from intrathecal administration for ASO drugs. If it does the nanoparticles through FUS becomes viable, would that need a lot less of the drug as well? So would it make it, probably could make it a fair bit cheaper as well?
Yeah, I mean, that's what we're thinking. So we would still need to provide repeat doses, but you know, we would probably, as you say, need a lot less drug, because we're delivering it to the site that it needs to act. And you know, that we can also sort of, I didn't mention this in the talk, but we can also sort of target particular cell populations. So we know that we need to reduce these protein aggregates in motor neurons, but we also know that microglia and astrocytes play a role in disease progression. And we can sort of target these therapies using a homing device on the surface of these nanoparticles to target particular cell types, and ensure that we get specific delivery to those cell types as well. So when you are using targeted therapy like that, by and large, you need less drug overall, which would probably drive the price down, as you suggest, Gethin.
Great. All right. Well, let's hope well, first of all, let's see if the ASOs work, but baby steps, and then it'd be great if we get better delivery mechanisms as well. Okay. Well, thank you both. That was some fantastic insights there. It always amazes me how good you guys are at translating horribly complicated science into bite-sized chunks for